Hello student, now we are going to discuss GIT Physiology. In the first chapter, we are going to discuss various motility of the GIT. In the second chapter, we will be discussing various hormones of the GIT, and in the third, various secretion and absorption at the level of GIT. So, first we are going to start with the functional anatomy of GIT. So, if you look at the GIT, so it's a basically tubular structure starting from the oesophagus up to the level of anal canal, right? This whole structure, okay, there are regional variation from different parts to different parts, but the overall theme of the GIT, if you look at, then it consists of 4 different layers, and all of you know that those layers are known as the mucosal layer of the GIT, submucosal layer of GIT. Below that, there will be muscularis layer or the muscle layer of the GIT, and on the surface, there is a covering layer that is known as the serosal layer of the GIT. So now, little bit more detail about the different layers. So first, the mucosal layer. So I am drawing a schematic diagram. Suppose this is the lumen of the GIT. Then this is the basement membrane of the epithelium cell. Onto that, you are going to find out columnar type of epithelium cell, okay, in the most part of the GIT. So this is the epithelium cell layer. I am writing here epithelium cell layer. Below the epithelium cell layer, you are going to find out a loose connective tissue cell layer. So this is the loose connective tissue only, okay. So this is known as lamina propria, right? Lamina propria. Below that, you will be finding out little, very thin layer of smooth muscles, right? So these smooth muscles are known as muscularis mucosae layer. Muscularis mucosae, okay. So this is basically the muscle layer of the mucosa. Why mucosa? Because all of you know that the combination of these 3, that is the epithelium cell layer, lamina propria and the muscularis mucosae, combination known as the mucosal layer of the GIT, right? Below the mucosal layer, obviously there will be the submucosal layer. Submucosal layer means it is again consist of loose connective tissue only. So this is the loose connective tissue layer, which is nothing but the submucosal layer. Submucosal layer. Now, if you look at the submucosal layer a little bit more, then you are going to find out that it contains large blood vessels. So the blood vessels, large blood vessels are present at the level of this submucosal layer. Not only that, various glands of the GIT, the large glands of the GIT, they are also present within this submucosal layer only. So the big blood vessels and the big glands of the GIT are present within this submucosal layer. Not only that, you have to remember that there is a neuronal plexus which is located at the level of submucosal layer also. So neuronal plexus means what you are going to find out in that plexus? You are going to find out multiple ganglions, and in between ganglions there are multiple neuronal collections. So this is known as the neuronal plexus. So what I am drawing here, so this is the multiple ganglion, and in between ganglia there are connection, okay, interconnection. So this is known as the neuronal plexus, which is located at the level of submucosal layer. That's why this neuronal plexus is known as submucosal neuronal plexus or Meissner's plexus. So this is more commonly known as the Meissner's plexus, right? So this is known as Meissner's plexus or submucosal plexus, right? Then after this submucosal layer, so this is the whole submucosal layer of the GIT. Below the submucosal layer, you are going to find out the muscle layer, the smooth muscle layer of GIT, which is responsible for the various motility of the GIT. Now these muscle layers, they are mainly 2 big layers, that is the inner circular muscle layer. So this is the smooth muscle which is present circularly at the periphery of the GIT. So that's why this is known as the circular muscle layer. And then there will be the longitudinal layer of the muscle. So this is basically the longitudinal layer of smooth muscle. So the combination of this circular and longitudinal smooth muscle layer is known as the muscularis propria or muscle layer of GIT, right? Now, in between this circular muscle and outer longitudinal muscle, there will be presence of another neuronal plexus here. There will be presence of another neuronal plexus here. So, if I draw another neuronal plexus, so ganglia, in between ganglia there is neuronal connection. So, this is ganglia and in between neuronal connection. So, this is neuronal plexus. This plexus is known as myenteric plexus, which is also known as Auerbach's plexus. Okay, Auerbachs, Auerbach plexus or myenteric plexus, right? So this is the muscle layer, and in between muscles the Auerbach plexus or myenteric plexus is located. And on the surface, I told you already, you are going to get a squamous cell layer. This squamous cell layer is nothing but known as the serosal layer of the GIT. So this is the GI serosal layer. So this is the overall structure of the GIT, the overall theme of the GIT, that there are 4 layers, and that is known as the mucosal layer of the GIT, submucosal layer of GIT, muscle layer or muscularis propria, and the serosal layer of the GIT. Now, in various parts of the GIT, this mucosal layer, they thrown into different folds. So what I'm talking about, I'm talking that this mucosal layer of the GIT in various parts, it's thrown into folds, or I can say there is exvagination and invagination. So evagination and invagination of this mucosal layer is also seen in different parts of the GIT. So this evagination part is known as the villi, right? All of you know this, villi. So obviously I can understand that because this is the mucosal fold, so onto the top of this mucosal fold, I am going to get my epithelium cell. Okay, so these are the epithelium cell. And at the top of the epithelium cell, you know, there will be presence of various microvilli, right? So this mucosal fold, which is forming this evagination, right? So this is nothing but the villi. On the surface of this villi, there is epithelium cell, and on the top of the epithelium cells, there will be microvilli. Now, in between, there will be presence of invaginations of this mucosal fold also, and this is known as the crypts. This is known as crypts of Leiber-Kuhn. So this is known as crypt of Leiber-Kuhn, right? So if you look at this crypt, right, what you are going to find out, at the bottom of this crypt, you are going to find out one cell which is known as the Paneth cell. Very, very important for MCQ. So at the bottom of this crypt, the Paneth cell is located. On both sides of this Paneth cell, you are going to find out a columnar base cell. This is known as columnar base cell. This columnar base cell is the stem cell of the GIT. So very, very important. Okay, you must have to remember. So at the bottom of the crypt, okay, or at the base of the crypt of Leiber-Kuhn, you are going to find out Paneth cell. Just above the Paneth cell on both sides, there will be columnar base cell, which is the active stem cell of the GIT. And then again you are going to get the Paneth cell. So this is again the Paneth cell, and again you are going to get the columnar base cell, okay, which is the stem cell. On the surface of this active stem cell layer, you are going to find out the proliferating stem cell layer, which is also known as the progenitor stem cell layer. So these are the progenitor stem cell layer, right? So these are progenitor stem cell. I am writing progenitor stem cell or proliferating stem cell layer. From these progenitor stem cells or proliferating stem cell, what will happen? There will be proliferation and differentiation. Because of the proliferation and differentiation, what will happen? This cell will ascend in the upward direction, right? And this proliferated and differentiated cell, they are ultimately going to form the epithelium cells of GIT as well as various other types of cell, like for example the goblet cells of the GIT. Getting my point? So they will differentiate and proliferate and they will ascend on the surface and they will form the epithelium cell. Similarly, this progenitor stem cell can also proliferate and differentiate and it can give rise to this Paneth cell also. So the bottom line of this thing is that the stem cells, the main stem cell or the active stem cells are located just beside the Paneth cell. That active stem cells is giving rise to the progenitor cells, and the progenitor cells are giving rise to various all other cells of GIT, starting from the epithelium cells as well as the Paneth cell, right? So this Paneth cell, which are located at the base of this crypt of Lieberkühn, they are important functionally. Why? Because it secretes a substances and that is known as the defensin, right? So that is known as the defensin. So it has some antibacterial property. Not only that, it also secretes lysozymes. Okay, lysozymes. Not only that, angiogenin 4 are also secreted by this Paneth cell. So at least you have to remember defensin, which is an antibacterial substances which is produced by this Paneth cell. Not only that, lysozymes are also secreted by the cells and angiogenin 4 are also secreted by the cell, right? So this is the overall structure of the GIT, different layer and then villi and the crypt. Now, I have already discussed that there are two neuronal plexus at the level of various layers of the GIT. These neuronal plexus, they contain huge number of neurons, okay, and it forms a nervous system within the GIT, and that is known as the enteric nervous system. So what I am saying, the enteric nervous system consists of two neuronal plexus. One is known as the Meissner's plexus, okay, and the other one is known as myenteric plexus, or Orbach's plexus, right? So this is part of enteric nervous system. In some of the books, they are saying the enteric nervous system is the third part of autonomic nervous system. We know that autonomic nervous system, that is nothing but sympathetic and parasympathetic division. Some books are saying that enteric nervous system is the third division of autonomic nervous system, and it is located only within the layers of the GIT. That's why this is known as enteric nervous system. So Meissner's plexus, I told you that this is located at the layer of submucosa. So if I ask you what is the main functions of this Meissner's plexus, so submucosal layer, look at this diagram. I told you that at the level of submucosal layer, there are big glands of the GIT and there are big blood vessels of the GIT. So the regulation of these blood vessels and these glands means regulation of the blood flow and the glandular secretion that is done by the neuronal plexus which is located close to them. And that's why we all know that this Meissner's plexus, they are responsible for regulation of the blood flow of the GIT as well as regulation of the secretion of the GIT. These two are the major functions of the Meissner's plexus. And Meissner's plexus is located from mid portion of the stomach up to the level of small intestine, right? So this is located from mid part of the stomach, up to the level of small intestine, right? Up to the level of small intestine. But if you look at the myndric plexus, it is located through and through the GIT, starting from the esophagus up to the level of the anal canal. And the myndric plexus is located in between two smooth muscle layer of the GIT, that is the circular and longitudinal smooth muscle. So what this plexus is going to do? This plexus is going to regulate the smooth muscle contraction by this way. Myndric plexus or Orbach plexus is responsible for regulation of motility of the GIT, right? So this is the main plexus which is responsible for regulating the motility of the GIT, and it is present throughout the GIT. Okay, it is present throughout the GIT. So this way you have to remember that enteric nervous system, it's an autonomic nervous system present within the layers of the GIT. If you look at the numbers of neurons within this enteric nervous system, that is huge, that is 100 millions neurons are present within the enteric nervous system. And the enteric nervous system is a self-sufficient autonomous system. It can functions normally without the help of, without the help of sympathetic and parasympathetic system. So although the sympathetic and parasympathetic system has effect on this enteric nervous system, but even if you cut this sympathetic and parasympathetic system, enteric nervous system can autonomously functions. It can maintain the all functions of GIT without any impairment. And that's why we say that enteric nervous system is like a brain, right? If you look at the numbers of neurons, this numbers is very huge, which is similar to the total population of neurons at the level of spinal cord. And because of this huge numbers of neurons, the enteric nervous system is an autonomous system, because of which this self-sufficient autonomous system is known as little brain or mini brain of the GIT, little brain of GIT or mini brain of GIT. It can function like a brain within the GIT, right? So this is all about your enteric nervous system. Now this enteric nervous system developed by neural migration, okay, from the neural crest, and that migration required the help of one gene, and that is known as the RET. So this rate proto-oncogene is responsible for the neural migration and the development of the enteric nervous system. This rate proto-oncogene, it is responsible for synthesis of tyrosine kinase receptor, right? So this receptor, this gene is coding a protein which is nothing but tyrosine kinase receptor. If there is mutation of this gene, then the neural migration will be defective and there will be a very severe type of congenital disease which all of you know, that is known as Hirschsprung disease, right? So this is known as Hirschsprung disease, congenital disease of the newborn, where the enteric nervous system, that is the myndric plexus as well as the Meissner's plexus are absent in GIT. Absent, okay. Which part of the GIT is mainly going to be affected? The colon, mainly the recto-sigmoid junction or part of the colon is going to be affected. But it is said that any part of the colon, including the small intestine, can also be affected. Including the small intestine can also be affected by the disease, where the enteric nervous system will be absent. So Why the enteric nervous system will be absent? Because of the defective neural migration. And if this enteric nervous system is not there, then the motility will be hampered and the newborn will suffer from severe constipation and the proximal dilatation of the colon. That's why this Hirschsprung disease is also known as congenital megacolon, right? So you have to remember these two important information, that absent ganglia due to defective neural migration. The rectosigmoid region is the main region where this ganglion are absent, but any part of the colon, even the small intestine, can also be affected by this disease condition, right? Now, apart from this enteric nervous system, we are again going to look at this structure, and we have to understand that GIT system is an open system. It is an open to environment, and constantly the GIT is getting various kind of antigen, various kind of external infection or something like that, that is constantly entering in our GIT. So GIT require a specialized immune system which will protect the enteric system completely, and that is known as enteric immune system, right? So if you look at the enteric immune system, right, it consists of various structure which is known as GALT, gut associated lymphoid tissue, gut associated lymphoid tissue, which is also known as MALT, that is mucosal associated lymphoid tissue. So from the name I can understand that this immune system is located within the mucosal layer of the GIT, and mucosal layer means consists of epithelium layer, lamina propria and muscularis mucosae. So this enteric immune system consists of, right, Peyer's patch, and it also consists of various population of lymphocyte, okay, as well as mast cell, and the third component is various lymph nodes of mesentery. So this enteric immune system has three major component. Peyer's patch. So what is Peyer's patch? All of you know that this is nothing but lymphoid follicles, right? So this is nothing but lymphoid follicles located at the lamina propria layers. So I'm just saying that at the level of this lamina propria, you are going to find out certain follicles. This follicle consists of B lymphocyte and T lymphocyte. The main follicle, the follicle is mainly consists of the B lymphocyte and little bit T lymphocyte also, and few dendritic cells are also there. But this follicle, as you can see, that this is aggregates of lymphocyte only, and little bit of dendritic cells. So the lymphoid follicles which are located at the level of lamina propria are known as the Peyer's patch. Now I told you that apart from this Peyer's patch, various different types of lymphocytes are also present. This lymphocyte can present in between epithelium cell of the GIT. So that is known as the intraepithelial lymphocyte, and some of the individual lymphocyte and mast cell can also be present within the lamina propria region. So within this lamina propria region, some lymphocyte will also be present, some mast cell will also be present, and some of the lymphocyte will be present in between this epithelial cell also. They are known as the intraepithelial lymphocyte. Now, above the Peyer's patch, if you look at the surface of this Peyer's patch, there is a specialized cell which is located in between the epithelium cell of the GIT, and this cell is known as the M cell. M stands for microfold cell of GIT. Microfold cells functions like antigen presenting cell. It functions, the function of this microfold cell is similar to the antigen presenting cell. How? So, suppose there is presence of certain antigen at the level of this lumen. I told you that GIT is constantly exposed to various antigens, various pathogens. So, if this is an antigen here, what will happen? This antigen basically is a protein one. This protein or this antigen will be directly taken up by this M cell by formation of some vesicles. Okay, so there is a process which is known as the clathrin mediated endocytosis. By the process of this clathrin mediated endocytosis, this whole antigen will be taken up within this M cell inside a vesicle. Then this M cell is not going to digest this protein. Okay, rather than this protein will be secreted from the basolateral membrane of these M cells, and the protein will be directly delivered to the level of the Peyer's patch. So what the M cell is doing? It is just taking the whole sample of the antigen and then it is delivering that antigen to the Peyer's patch. Why? Because Peyer's patch consists of B lymphocyte and T lymphocyte. So basically antigen presenting cell like function, but M cell is not exactly the antigen presenting cell because it is not digesting the antigen. Because you know, the antigen presenting cell, they also digest the antigen and then they express the most immunogenic part of the antigen. But here it is not digesting because it lacks various lysosomal enzyme. And what it is doing, it is directly taking up the protein and then delivering the protein to the level of Peyer's patch. So it is asking the Peyer's patch to handle the antigen rather than self-handling. That's why I told that M cells behave like an antigen presenting cell, but it is not exactly the antigen presenting cell, right? So this way you have to remember the functions of Peyer's patch and the entry immune system functions, right? So this is briefly about the gut associated lymphoid tissue or mucosal associated lymphoid tissue. Now after discussing this structural part, now we are going to discuss the actual motility of the GIT. So if you classify the various motility of the GIT, you know that we classify like this. Number one is the electrical motility of the GIT, and number two is the mechanical motility of the GIT. Right. Under electrical motility of the GIT, we are going to discuss BER. BER stands for basal electrical rhythm of GIT or basic electrical rhythm of GIT, right? So this is an electrical activity in the GI smooth muscle. We are going to discuss in detail about this basal electrical rhythm. Now coming to the mechanical motility, you know that mechanical motility varies depending on whether the food is present in the GIT or not. So the fed state motility and the fasting stage motility are different. Fed state motility in the GIT again classified into peristalsis and segmentation, right? So peristalsis motility and segmentation motility. And the fasting motility is very important for your MCQ, that is known as MMC. MMC stands for migratory motor complex, right? This is migratory motor complex, right? So peristalsis, segmentation and migratory motor complex, these are the major types of motility in the GIT. Apart from that, you are going to find out there is a special type of peristalsis at the level of large gut, which is known as mass movement. We are also going to discuss little bit about this mass movement, which is only seen at the level of colon or large gut, right? So peristalsis, segmentation, MMC and mass movement. If I ask you which one is the dominant motility in fed state of GIT. Obviously, MMC is a fasting motility, but if I have to choose between peristalsis, segmentation and mass movement, which one is the dominant and predominant motility in fed state, your answer will be segmentation, not the peristalsis. Answer will be segmentation. So this is the dominant motility in fed state of GIT. You will be understanding why this is the dominant motility, not the peristalsis, right? So first we are going to discuss this electrical motility of the GIT, that is the BER, and then we are going to discuss briefly about these different other types of motility in GIT, right? So we are starting with the basic electrical rhythm or basal electrical rhythm of GIT, BER. If I ask you, tell me in one word that what is BER or two words, your answer will be BER is nothing but resting membrane potential of GI smooth muscle. So remember this, BER is nothing but RMP of obviously muscles, GI smooth muscle, right? So this is the bottom line that you have to remember in your brain. BER is very confusing, but it is very important for MCQ. So BER is nothing but RMP of smooth muscle cells of GIT, smooth muscle cells of GIT. Now, if you look at the RMP of other tissue, like for example skeletal muscle or myocardium, then it is a fixed membrane voltage, like for example skeletal muscle minus 90, neuron minus 70, myocardium minus 90. But GI smooth muscle RMP is a little different. So we are going to plot this BER or the RMP of the GI smooth muscle on this x-axis, which is the time axis, and on y-axis, which is the membrane voltage axis. You will be finding out that the membrane voltage or the resting membrane of the GIT, it is moving from minus 65 millivolt to minus 45 millivolt, and again it is returning back to minus 65 millivolt, and again it is rising to minus 45 millivolt, and again it is returning back. So what we are finding out here, that the GI smooth muscle RMP is not a fixed voltage, rather the electrical voltage is fluctuating, is undulating from one voltage, that is from minus 65 to minus 45 millivolt. This undulating type of RMP, this constant fluctuation of the RMP from minus 65 millivolt to minus 45 millivolt is known as the basic electrical rhythm or basal electrical rhythm or RMP of the GIT. So I can find out that the RMP of the GI smooth muscle, it is fluctuating in a rhythmic fashion, even in the resting condition of the GIT, and that's why this is known as a basic rhythm, basal rhythm, right? Now, why this rhythm is fluctuating, why it is not a fixed voltage? This kind of fluctuation voltage we have also seen at the level of SA node of heart, and we have seen the SA node membrane voltage was fluctuating because it contained the pacemaker cells. So is there any pacemaker cell at the level of GIT which is generating this basic electrical rhythm or BER? Answer is yes. So if I ask you why the BER is originating, so what is the origin of the BER, your answer will be that this is due to presence of pacemaker cell of GIT. All of you know that the pacemaker cell of GIT is known as interstitial cells of Casals, right? So this is known as interstitial cells of Casals or Cahals, right? So these interstitial cells of Cahals or Casals are very important for your MCQ. Remember that this is the cell which is located in between the smooth muscle layers of the GIT. So suppose this is the circular smooth muscle, okay, and this is the longitudinal smooth muscle. In between these two smooth muscle layer, you are going to find out these interstitial cells of Cahals. You are going to find out these interstitial cells of Cahals. Within these interstitial cells of Cahals, there is spontaneous fluctuation of the membrane voltage due to presence of certain channels which is still not confirmatory. But because of the constant fluctuation of the membrane voltage within these interstitial cells of Cahals, the membrane voltage of the smooth muscle also fluctuate from minus 65 millivolt to minus 45 millivolt, and this is nothing but basic or basal electrical rhythm. Okay. Also remember that these interstitial cells of Cahals, they are also located at the level of kidney. Where in the kidney? At the level of pelvic ureter junctions. And the contraction of this ureter is due to spontaneous production of impulses from the interstitial cells of Cahals, which are located at the pelvic ureter junctions. That has already been asked in AIIMS MCQ, so that's why you have to remember this extra information. Not only that, you also remember another information that the C-kit is the marker of these interstitial cells of Cahals. The scientists are isolating these interstitial cells of Cahals by identifying the C-kit. That's why the C-kit is the marker, which is a proto-oncogene, all of you know, is a marker of these interstitial cells of Cahals, right? Now, what is the location of these interstitial cells of Cahals? So, the interstitial cells of Cahals, they are located from the mid part of stomach. They are located from the mid part of stomach up to the level of anal canal, right? But they are not present at the level of esophagus or even not present at the level of fundic and cardiac end of the stomach. They start from the mid part of the stomach, somewhere in the body region of the stomach, and then they are present throughout the other part of the stomach, as well as small intestine, as well as large gut, as well as up to the level of anal canal, right? So you have to remember that where these interstitial cells of Cahals are located. So these interstitial cells of Cahals are basically giving rise to this BER or basal electrical rhythm. In different part of the GIT, if you look at the rate of this BER, so if I calculate what is the rate of BER, you are going to find out that at the level of stomach, the rate of BER is 4 per minute, right? So with the help of this x-axis, that is the time axis, if you find out that how many peak, how many waves you are getting in 1 minute, then at the level of stomach smooth muscle, the rate is 4 per minute. At the level of duodenum, the rate is maximum, that is 12 per minute. At the level of jejunum, the rate is 11 to 12 per minute. I am writing 11. At the level of ileum, right, the rate is 8 per minute. At the level of cecum, the rate is very minimum, that is 2 per minute. And at the level of sigmoid colon, the rate is 6 per minute. Believe me, these rates of BER is very important. Multiple times various MCQ has been given from this rate. So the minimum rate from this number I can find out the cecum, and the maximum rate at the level of duodenum. But you have to remember all of this rate because you don't know which options will be given in your MCQ. So that way, remember the rate of BER. Now what the BER is doing? Can BER produce muscle contraction or motility? Answer is no, because BER is like a resting membrane voltage. Can resting membrane voltage causes muscle contraction? Answer is no. You require action potential. So BER per se cannot produce muscle contraction. But suppose there are certain kind of stimulation. Like for example, if I give a little bit of acetylcholine, what will happen? This BER will rise, rise, and then it will go above minus 45 millivolts. So whenever it is going above this minus 45 millivolt, this 45 millivolt is the threshold voltage. This 45 millivolt is the threshold voltage of the GI smooth muscle. So in presence of certain stimulus, like for example acetylcholine, the BER is crossing the threshold, and whenever it is crossing the threshold, we all know that action potential is going to be generated. Multiple action potential will be generated at the top of this BER. This kind of action potential are known as the spike potential. Please remember, whenever there is generation of the spike potential, there will be contraction of the smooth muscle of this GIT, and this contraction will leads to various types of motility of the GIT. So getting my point. So in resting condition, continuous membrane voltage is fluctuating. That is not contracting the muscle. Motility is not seen. But whenever you are giving certain kind of stimulus, in for example acetylcholine, this BER is crossing the threshold and then multiple spike potential are generated. That is causing muscle contraction, and that will produce various motility of the GIT, like for example peristalsis, segmentation, etc. Okay, so this is all about the BER. Now you look at the graph which is given in genome. So this is the graph which is given in genome. Okay, look this graph carefully. So first of all, you are finding out that membrane voltage is fluctuating. So simple fluctuation of the membrane voltage is nothing but BER. Okay, so this is nothing but my BER. Okay, when BER is occurring only, muscle contraction is not going to happen. So if you measure the contractile strength of the muscle or tension, you are finding out that there is no tension. The tension is equal to 0. Tension is 0. No contraction. But whenever you are giving cholinergic kind of stimulation, acetylcholine. So acetylcholine from this window up to this window, what you are finding out, the BER is crossing the threshold, and at the top of this BER you are finding out spike potential. These are also spike potential. Whenever there are spike potential, what is going to happen? This will leads to contraction of the muscle, and contraction of the muscle means if you measure the tension, then you are going to find out that the muscle tension are rising. That's why we say that in presence of cholinergic stimulation, there will be contraction, okay, of the smooth muscles of the GIT, because BER will cross the threshold. But rather than giving cholinergic stimulation, if you give adrenergic stimulation, like for example in presence of epinephrine and norepinephrine, what is going to happen? The BER will reach below the threshold voltage. So as you can find out here that the BER not able to reach up to the level of threshold. So what is happening? The spike potential are disappearing, and whenever the spike potential are disappearing, what is going to happen? There will be again relaxation of the smooth muscle, and relaxation of the smooth muscle means the tension is going to be reduced. So this way you have to remember this curve. This is very important, that how the BER is changing in presence of various neurotransmitter, like for example acetylcholine, epinephrine and norepinephrine. Now suppose I have given this graph as an image based question. I told you that identify that which part of the GIT has been given in this diagram. So what you are going to do, you are going to count the rate of BER. How to count? So this is the 10-second window. Within this 10-second window, you are getting only one peak of the BER. So within 10 second, 1. That means within 60 second, how many I am going to get? I am going to get 6. So the rate of the BER according to this image is 6 per minute. 6 per minute BER rate means which part of the GIT you have isolated, as you can find out from here. that the sigmoid colon has a rate of 6 per minute. So by this way, you can identify from the BER that the which part of the GIT has been isolated for the experimental purpose, right? So this is all about the BER. You look at MCQs, okay, regarding this BER only. So first MCQ, slow waves or BER maximum in. So BER is also known as the slow wave of the GIT. So maximum in. So if you look at the options, the stomach 4 per minute, ileum 8 per minute, colon, okay. So colon and rectum, they are the 6 per minute. Okay. So what you are finding out, that maximum rate is at the level of ileum. So your answer is ileum or ileum here. Then second question they are asking, that pacemaker of the GIT is located in which part? I told you already that pacemakers start from the meat part of the stomach, that is the body of the stomach. So cardiac end of the stomach, pacemaker or the interstitial cells of Cahals are not there. Pyloric end of the stomach, it can be present here. Central control of CBD in origin, obviously this is a false option which is given, which is not related to this. And fundus of the stomach, I told you already that cardiac end as well as fundus end of the stomach, there is no pacemaker interstitial cells of Cahals. So only and only answer that is possible here is the pyloric end of the stomach, right? So these are the different types of MCQ which can be given from the BER. Now we are going to discuss the next motility, that is the peristalsis of the GIT. So all of us know that peristalsis is known as the propulsive motility of the GIT. Okay, and the speed of peristalsis, if you look at the speed of peristalsis, then it is 2 to 25 centimeter per seconds. So look at what is the speed of peristalsis. This is very high, 2 to 25 centimeter per seconds. If peristalsis is the dominant motility after food intake, what is going to happen? Whenever you are taking food, the peristalsis is the dominant motility. It will move your food particle with a rate of 25 centimeter per second. What is going to happen? After a few minutes, the food will come out of the anal canal. But that is not going to happen, you know. So, what is the dominant motility in fed state of the GIT? I told you segmentation, not the peristalsis. Right? Then peristalsis, it is seen in which part of the GIT? It is seen in throughout the GIT, starting from upper end of the esophagus up to the level of the anal canal. Now, in this diagram, you can find out that this is the food bolus. We all know peristalsis means proximal to the food bolus, there will be contraction of the GIT. And ahead of this food bolus, there will be relaxation of the GIT. So what will happen? Whenever the food particle is stretching in any part of the GIT, the proximal part will contract and the distal parts will relax and the food particle will move in forward direction. So that's why we say that peristalsis is a kind of propulsive motility where food particle always move from proximal to the distal end of the GIT. Now there are certain MCQ regarding the mechanism of the peristalsis. So how peristalsis occurs? Okay, so suppose this is the food bolus which is present at the lumen of the GIT. In presence of food bolus, what will happen? There will be stretch on the GIT. So this food bolus is causing stretch of the GIT. Whenever there is stretching of the GIT, okay, what happens? The epithelium cells which are present, they are going to secrete one neurotransmitter, and that is nothing but serotonin. This serotonin will stimulate the afferent neuronal ending which are present at the mucosal layer of the GIT. So this is the neuronal ending which are present at the level of mucosal layer. Now what this neuronal ending will do, these are the afferent, means it will bring the sensation to the level of the ganglia of the GIT, right? So this ganglia is part of the neuronal plexus of the GIT, which we have already discussed that there are two neuronal plexus, Meissner's plexus and Meinrich plexus. So whenever the afferent input is coming from the mucosal layer up to the level of this ganglion of the GIT, now this ganglion will send signal, okay. Certain signal will go to the distal part, towards the distal part of the GIT, and certain signal will go towards the proximal part of the GIT through the neurons. Now the GIT neurons are arranged in such a way that the neurons which are excited towards the proximal ends of the GIT, they are going to secrete neurotransmitter which is nothing but acetylcholine and substance P, right? So the neurons which are excited in the proximal part of the GIT, they are going to secrete two neurotransmitters, acetylcholine and substance P. We all know that in presence of acetylcholine and substance P, what is going to happen to the smooth muscles of the GIT? There will be contractions. So that's why the proximal to this food particle, the GIT will contract. But if you look at the neurons which are projecting towards the distal part of the GIT, they are going to secrete other neurotransmitter like, for example, vasoactive intestinal polypeptide, VIP, nitric oxide and adenosine triphosphate. In presence of this neurotransmitter, we all know that there will be relaxation of the smooth muscles of the GIT. That's why the distal part of the GIT ahead of this food particle is going to relax, and this is nothing but the peristalsis. So now I can conclude here that the proximal contraction in case of peristalsis is done by acetylcholine and substance P, and distal relaxation is done by VIP, nitric oxide and adenosine triphosphate, right? So this is all about the mechanism of the peristalsis, and this is brief discussion of peristalsis which is required for your MCQ. Now I told you that there is a special type of peristalsis which is only seen at the level of large gut, that is a colon, right? So that's why it is written here that mass movement is a special kind of peristalsis which is seen only at the level of colon. You must have seen that just after taking food, huge amount of food, what you have seen that there is a pressure for defecation. This pressure originates because of the mass movement at the level of colon, right? So that's why it is said that the mass movement is initiated by gastrocolic reflex or duodenocolic reflex. Gastrocolic, so whenever the food particle is entering at the level of stomach, the colon is start moving, the colon is starting its motility, or when the food is present at the level of duodenum, the colon motility increases. That's why this is gastrocolic and duodenocolic reflex, which is nothing but initiator event of the mass movement. This mass movement is very, very prominent in case of infant, and that's why you must have seen that just after meal, right, the infant defecates. Okay, why? Because the prominent mass movement is seen in case of infant, and whenever they takes even the small amount of food, that food will start the gastrocolic and duodenocolic reflex, and that will initiate the mass movement, and mass movement means the defecation may occur, right? So that's why it is written that initiated by gastrocolic or duodenocolic reflex. It can also be initiated by distension of the colon also. So if you introduce any probe in the colon and if you dilate or stretch the colon, then also the colonic motility enhances, right? That can typically seen during the colonoscopy, right? Okay. And obviously, the motility means it is nothing but controlled by Ortsback plexus or Meinrich plexus. So if you causes distraction of this Ortsback plexus, then obviously the mass movement will be abolished. Now there is a particular MCQ that has been asked in AIIMS entrance examination, that maximum, okay, mass movement is seen in which part of the colon? Okay, so mass movement means I can say that this is nothing but a postprandial motility mainly. Postprandial means just after food, the motility which is enhanced at the level of colon, that is nothing but the postprandial motility, or I can see that this is nothing but the gastrocolic or duodenocolic reflex. So they are asking that this gastrocolic or duodenocolic reflex is maximally seen in which part of the colon? Your answer is sigmoid colon, followed by transverse colons, right? Followed by transverse colon. So maximum movement of colon which is seen after food is nothing but at the level of sigmoid colon and followed by transverse colon, right? There is a controversy regarding this MCQ, so please be careful. The answer is sigmoid colon, maximum postprandial motility. So this is all about mass movement. So peristalsis and mass movement is done. Now look at the other movement, that is the segmentation movement, or the major or the predominant movement in fed state of GIT. So segmentation. If you look at this diagram, okay, you have to look at this diagram carefully to understand the segmentation movement. So segmentation movement means within a part or within a segment of the GIT, the food bolus will move in to and fro direction. The food bolus will move like a pendulum of clock from this side to this side, but there will be no net movement of food particle from one part to other part of the GIT. The food will remain within the same segment of the GIT. That's why within the same segment, the motility or the movement is known as the segmental motility or segmentation. So if this is the particular segment of the GIT, suppose I have taken this particular segment, what I am finding out, look at this diagram, okay? So I can find out that within this segment, some part of the GIT is contracted. So this is these are the contractile ring, and in between the food particle is present here. Now what will happen after certain point of time, the area where contractile ring were there, this area is going to be dilated, and the area where there was dilatation, this area is going to be contracted. What I am saying, look at. So now I can find out the contractile ring will appear here, and okay, so the contractile ring will appear here, okay, this part will dilate, and again the contractile ring will appear here, right? I hope you are understanding this part. Okay, so this was contracted, now this is dilated, right? And the area which was dilated, now this is contracted. Okay, again this area was contracted, so this part will be dilated, and the area which was dilated, this part is going to be contracted like this. So if I draw a diagram, you will find out that. So this is the same segment of the GIT, but now I can find out the different types of contraction. So what will happen, whenever the area where the food bolus was there, if this area is getting contracted, so obviously this food bolus will be divided into two parts. Some part of the food bolus will move in the proximal direction, and some part of the food bolus will move in the distal direction. Again, in the next minute or in the next time, what will happen? Again the same thing, the part which was dilated, now this part will be again contracted like this, okay, and the part which was contracted, they are going to be dilated like this, okay. So again the similar kind of picture. So this is similar to this first picture. So what will happen, whenever the part which was dilated, if it is getting contracted, so again it will push the food bolus in the distal part, and whenever this part is contracted, it will push the food bolus in the proximal part. So again these two food bolus is coming close to one another, and they will mix up here, right? And again the same kind of movement is going to happen, because of which I can understand that the food particle is not moving from one part to the other part of the GIT. There is no net movement of the food particle, but the food bolus or the food particle is remaining within the same segment, but it is moving from one part to another part, again to the same part, again and again, to and fro movement, pendular movement. Okay, this kind of movement is known as the segmentation movement of GIT. So what is the role of this segmentation movement? Why this is required? Because you are not pushing the food from one part to other part. So whenever segmentation movement is happening, I can understand that the food bolus is crushed into multiple small fragment. So what will happen? Whenever the food particle is crushed into small part, the secretion of the GIT will be properly mixed up with this food particle, right? So the segmentation movement is giving some time, and it is helping the secretion to be mixed up with the food particle properly. Not only that, even after the digestion of the food particle, you require certain amount of time for the proper absorption of this food particle. So if peristalsis is the major motility, then food will move from one part to the other part of the GIT and the absorption time will not be available. But segmentation movement means it is keeping the food particle within the same segment. So we say that segmentation movements means it is providing time for proper digestion of the food particle as well as proper absorption of the food particle. It is providing time for proper digestion and absorption. How it is providing time for digestion? It is helping the mixing of the GI secretion with the food particle, and it is providing time for absorption. That's why we say that segmentation is more important than even the peristalsis also, because the major function of the GIT, that is the digestion and absorption, is based on this segmentation movement, right? So this is all about segmentation that you have to read for your MCQ. Now, the next major motility, which is again very important for MCQ, is the migratory motor complex or MMC. Migratory motor complex or MMC, right? So I told you the first important information that you have to remember is this is the fasting motility of GIT. So this is the fasting motility of GIT. This motility will be seen only and only when you are in a fasting stage, right? Now, MMC, where from this MMC is going to start? So start of MMC is seen from the mid part of stomach, and ending of this MMC is at the level of terminal ileum. Terminal ileum. So what I am saying, if you look at the whole structure of the GIT, suppose this is the stomach and then this is the small intestine, okay? So it will start from the mid part of the stomach and then it will ends at the terminal end of the ileum. Fasting motility, there will be a strong contraction of the GI smooth muscle which will move from this stomach mid portion of the stomach and it will go up to the level of this terminal ileum. Question is, why you require this kind of strong contraction when the food is not there at the level of GIT? Why you require fasting movement of GIT? See, whenever you are taking certain food particle, there will be digestion, there will be absorption. Even after complete digestion and absorption, certain amount of food particle, okay, small amount of food particle, small amount of various intestinal and gastric juices will remain even after complete digestion and absorption. Now I have to clear all of this remaining food particle, all of these remaining gastric juices which are still present inside the various part of the GIC. So I have to clear up all this part. Okay, why? Because I have to prepare the GIT for the next food. Okay, so this clearing of these remaining food particle, clearing of these remaining gastric juices, enzymes is done by strong contraction of this GIT, which will squeeze out all of the remaining content, and they squeeze out all of this content up to the level of this colon. So from the level of stomach, intestine, all of this content will be squeezed out up to the level of colon, and that is done by this MMC or migratory motor complex. So this is basically the sweeper of the GIT. It is basically clearing the content of the GIT. That's why we say that MMC is responsible for housekeeping function of GIT, right? So it is maintaining the clearing movement of the GIT, housekeeping function of GIT. Very important. Okay, so interdigestive period, in the interdigestive period the MMC is seen, right? So the question is, you are saying again and again that this is a fasting motility. So how much fasting is required for the start of this motility? So first time, if you are fasting for last 90 to 120 minutes, so it will start only after, okay, only after 90 minutes of last food. So, okay, 90 minutes of last food, right? So if your stomach is empty for last 90 minutes at least, okay, then and then only this MMC is going to start. Whenever MMC start, the strong contraction will move from this mid portion of the stomach up to the level of this ileum. You will feel a little bit contractile pain inside your abdomen. We know that is known as the hunger pain basically. Then this contraction will disappear. Again this contraction will reappear for second time after a interval of. So this will reappear at an interval of again 90 to 120 minutes. So it is appearing first time after the 90 minutes of the last food, and again it will reappear at an interval of 90 to 120 minutes. This movement will continue and it will get more and more stronger until and unless you take some food. Whenever certain kind of food is entering in the GIT, this MMC will disappear, right? If you look at the speed of this MMC, compare this speed with the peristaltic speed. So this is 5 centimeter per minute. So you can understand that peristaltic speed is 2 to 25 centimeter per seconds, but here 5 centimeter per minute means very slow movement, but very strong movement, hunger pain, right? MCQ has also been asked that who regulate the MMC. So please remember, regulation of the migratory motor complex is done by a hormone which is known as motility hormone. Motility hormone is secreted by a special kind of cells in the GIT which is known as MO cells. MO cells are located mainly at the level of duodenum, jejunum. It is also located at the level of stomachs and parts of colons also, but the main location of this MO cell is duodenum and jejunum. So this motility hormone is responsible for initiation as well as regulation of the migratory motor complex, right? So this is all about the fasting motility or MMC, right? So now we have discussed the major motility, the overall major motility of the GIT. Lastly, you have to read certain information about motility of the stomach or gastric motility. Okay, so you know that at the level of stomach, if you look at the proximal part of the stomach, there are two major motility. One is known as receptive relaxation of the stomach, receptive relaxation of the stomach, and other one is known as the adaptive relaxation of the stomach, right? So probably I have discussed this receptive relaxation at the general physiology chapter. What is receptive relaxation? Whenever the food particle is entering at the upper part of the esophagus, food particle is entering in the upper part of the esophagus. Okay, so at the upper part of the esophagus, food particle has just entered. The upper region of the stomach will dilate ahead of food particle. So food particle has not reached yet towards the stomach. It is just in the upper part of the esophagus, but stomach is preparing to receive this food particle. This kind of phenomena is known as receptive relaxation. So this food particle is dilating the upper part of the stomach. This is nothing but receptive relaxation. It is said that the receptive relaxation is a kind of feed forward mechanism, right? Feed forward mechanism. We all know that feed forward mechanism means it is an anticipatory control system where you are anticipating something. So the stomach is anticipating here that the food is going to come, so I have to accommodate the food particle. So what I will do? I am relaxing. So this is known as the receptive relaxation. The mechanism for receptive relaxation is done by vagus neuron. So this is mediated by vagus reflex pathway, and the actual relaxation is done by nitric oxide neurotransmitter, which is secreted at the level of the smooth muscle, right? Okay. Now, suppose the food particle has already entered into the upper part of the stomach. Now, again the stomach will dilate to accommodate this food particle which has already entered inside the stomach. So in presence of food particle, the stomach is relaxing to adapt with the food bolus. This is known as adaptive relaxation of the stomach. Adaptive relaxation of the stomach is regulated by local vagovagal reflex. Local vagovagal reflex. So there are various local reflex circuits are present within the stomach, and they regulate the relaxation of the stomach which is in presence of food particle. So this is adaptive relaxation. These two are very, very important, right? Receptive and adaptive relaxation. Now, in the lower part of the stomach, that is the antropyloric region of the stomach, you have to remember that there is a sphincter here which is known as the pyloric sphincter. Controlling the pyloric sphincter means if you cause contraction of the pyloric sphincter, what will happen? The food particle will not be able to move from the stomach to the level of duodenum. That means you are decreasing the motility of the stomach. And if you are causing relaxation of this pyloric sphincter, then the enhanced motility will be seen at the level of stomach, so that the food particle will easily move from stomach at the level of duodenum. So, what are the regulator of the motility of the lower part of the stomach, or what are the stimulus for pyloric sphincter control? Okay, so look at the gastric emptying or regulation of the pyloric sphincter is mainly done by various neural reflexes. This has been asked in MCQ. That's why I'm giving so much stress here, that gastric motility is mainly regulated by neural reflexes, right? Most rapidly emptying substances is isotonic saline. Suppose you are taking certain food, you are taking certain liquid, which will move faster from stomach to duodenum. Isotonic saline is the fastest, right? Then looking at the food particle, if you take 3 different types of food, you can easily find out here that carbohydrate will release from stomach to duodenum for the first time, followed by protein will take some more time, and fat will take very huge time to move from stomach to the level of duodenum. That's why you can understand that if you take fatty food, that food will remain inside your stomach for long duration, and you will feel the fullness inside your stomach after a fatty meal, right? Now, if you causes distension of the duodenum, so suppose the food particle has moved from the stomach to the level of duodenum, so duodenum is distended due to food particle, right? This will inhibit the pyloric sphincter. Not only the distension of the duodenum, the acidic content in the duodenum. So if the content, the stomach content is very, very acidic. So whenever that acidic content is entering in the duodenum, that will also causes contraction of the pyloric sphincter. Not only that, high or low osmolality containing food. So if the food osmolality is very, very high or very, very low, then also the pyloric sphincter will be contracted. This is mediated via a neural reflex which is known as enterogastric reflex, right? So the reflex is originating from the duodenum and it is inhibiting the pyloric sphincter. It is inhibiting the pyloric sphincter. So this is known as enterogastric reflexes. Okay, whenever there is distension of the duodenum or acidic content in the duodenum or very high osmolar food is present in the duodenum, it causes contraction of the pyloric sphincter. Apart from this, certain hormones you also have to remember, right? For example, gastrin, okay, stimulate the emptying of the stomach, means gastrin causes relaxation of the pyloric sphincter, okay, relaxation of pyloric sphincter. And the CCK, secretin and GIP, they inhibit the gastric emptying, means they causes contraction of the pyloric sphincter. Very, very important. I'm telling you, these are very, very important for your MCQ. Not only that, this information is also important for your MCQ. So this is all about gastric motility, and this finishes our motility chapter. But before that, okay, we are going to discuss certain MCQ before finishing. Gastric emptying is mainly regulated by, so direct question, I told you the neural reflex is the main one, right? Stimulation for gastric emptying, right? So distension, okay, or hormone. Which hormone? Gastrin is the hormone which causes relaxation of the pyloric sphincter and it stimulates the emptying of the stomach. But if you look at CCK, okay, it will inhibit the gastric emptying. Okay, distension of the duodenum will inhibit the gastric emptying. Okay, so distension of the duodenum, if it is duodenum only, but if it is distension of the stomach, so distension of duodenum will inhibit the pyloric sphincter, but distension of the stomach will secrete one hormone which is nothing but gastrin. This we'll discuss in the endocrine chapter, GI endocrine, right? GI hormone. This gastrin is a stimulus for pyloric sphincter. So basically, this D can also be an answer in this question. If you have to choose multiple option, then the gastrin is the direct stimulus for gastric emptying, and distension of the stomach is an indirect stimulus for gastric emptying. But if I have to choose only one answer, obviously I am going to choose gastrin here, because they have not mentioned the distension of duodenum or stomach. Assuming that this is distension of duodenum, the best or single best answer possible here is gastrin. Now next to MCQ, intestinal motility is increased by. So here it is not the motility of the stomach, intestinal motility, which I have not discussed, because detail of the intestinal motility you don't have to read. But remember, 3 hormones which enhances motility of the intestine, and they are number 1 CCK, motilin and gastrin. All these 3 hormones, they enhances the motility of the intestine. But if you look at the potency, then CCK is the most potent, followed by motilin, right, followed by gastrin. Okay, so that's why there is a chances that you are going to get mistake in this MCQ. You most probably are going to mark motilin as your answer, but CCK twice more potent than that of the motilin in respect to intestinal motility. So single best answer is CCK, right? And maximum postprandial motility, I told you postprandial motility means this is nothing but the gastrocolic reflex or duodenocolic reflex. The colon will start enhanced movement. The maximum is seen at the level of sigmoid colon followed by transverse colon, right? So this is all about your motility of GIT.